Welcome to Conversations on Ichnology. The International Ichnological Association is pleased to offer this lecture cycle to all those interested in ichnology. The lectures took place on the last Friday of each month. Thank you for joining us for the final lecture of this year. Also, thanks to our IAEA members for their support of our association. The IIA is committed to assisting as many students as possible in participating in IGNIA 2028, and more members are needed to accomplish this goal. At the beginning of this year, IIA provided new member categories that make memberships more accessible to all. Thus, we invite those who are not yet members to join our association. About today's talk, Dr. Andrea Baucon is a paleontologist at the University of Cagliari. He has investigated the potential of ichnofossils in astrobiology, both theoretically and practically, using predictive modeling to assess the ichnological potential of the Mars 2020 landing site. This astroichnological perspective is inspired by his study of ichnofossils in extreme environments, including chondrites in low oxygen settings, and the ichnological documentation of the earliest deep-sea vertebrates. Andrea also explores the humanistic aspects of ichnology, studying its history and engaging in science outreach. He has curated international exhibitions to promote ichnological heritage and manages social media channels dedicated to trace fossils. And in this opportunity, Andrea will share with us the talk titled Cosmic Traces, How Ichnology Can Guide Our Search for Extraterrestrial Life. Thank you very much, Andrea, for accepting this invitation.
So, hello, everyone. Thank you for inviting me to give this provocative talk. And I say provocative because the idea of applying ichnology to the search of extraterrestrial life holds a great potential, but it's not so widespread. The humanity is looking to the sky for thousands of years, and scholars like Giordano Bruno already in the Renaissance thought about possible life beyond Earth. But the search for extraterrestrial life is as old as humanity. Indeed, Greek researchers, like ancient Greek researchers, like Lucian of Samosata, were imagining travel on the moon and in other extraterrestrial locales. So, what have we found in thousands of years of searching for life beyond the Earth? Well, the answer is quite depressing. Until now, we did not find any extraterrestrial life form. Even if there are some promising results coming, for instance, from NASA, at present there is no convincing evidence for alien life. So there is a conflict between this absence of evidence and the argument that extraterrestrial life should be common in the universe. After all, it seems that our universe it's very favorable for life. This conflict has been synthesized by Enrico Fermi, an Italian physicist and Nobel Prize laureate, with a very catchy sentence. If the universe is teeming with aliens, where is everybody? I will try to tackle this question using life substrate interactions and their products that are ichnofossils like barrows, trails, borings, and even microbially induced structures. And specifically in this presentation, I will focus on 3 points. The first is why ichnofossils are important for astrobiology and the search of extraterrestrial life. Second, what have you found so far? And finally, we will explore which tools are best suited for ichnofossil detection. I will start from the first point, why ichnofossils are so important for the search of extraterrestrial life. And the most prominent aspect is the preservation potential of ichnofossils. Ichnofossils preserve very, very well. Apparently, body fossils like this trilobite and biochemical biosignatures are more robust evidence for eventual extraterrestrial life. However, body fossils and biochemical signatures have a relatively low fossilization potential. So that's why Matt Damon is so unhappy in the slide. Fossilization is a rare occurrence, so it would be not so easy to find chemical and body fossil signatures on other planets. Fortunately, ichnofossils come to rescue. And I can assure you that I did not pay Matt Damon for acting in these slides. Jokes apart, one of the best places in the world to acknowledge the preservation potential of ichnofossils is in Portugal, in Peña Garcia. Within the area of the UNESCO Geopark Naturtejo, there is a thick quartzite succession. Quartzite is a metamorphic rock, so don't imagine to find many body fossils here. However, trace fossils are quite abundant in Peña Garcia. And you can appreciate this from this beautifully preserved Cruziana. These are trilobite barrows produced in the Ordovicians. And even bioglyphs are very well preserved, despite the fact that this succession consists of metamorphic rocks. And one prime example of the ichnological potential of Peña Garcia is the largest trilobite barrow that, if I remember well, is about 29 centimeters wide. So the Peña Garcia trace fossils show that trace fossils may survive to processes like metamorphism and teknonism that would obliterate other signatures of life. Obviously, I don't expect to find trilobite barrows on Mars. But slime molds, which are unicellular amoeboid organisms from Earth, show us that even unicellular life can produce macroscopic traces. Like you can see, this sort of slug is made of many unicellular individuals that behave as an individual only when food is scarce. they aggregate to form a multicellular slug-like creature. So, coming back to Mars, we can imagine that if simple life existed on Mars, we could find trails and eventually burrows. But I should highlight that so far no evidence for life has ever been found on Mars or on other extraterrestrial bodies. The second reason why ichnofossils are important in the field of astrobiology is that they are universal biosignature. In fact, our big question, is there life beyond Earth, comes with a huge asterisk. That is, will we know extraterrestrial life when we see it? In other words, and this is shown us by science fiction, extraterrestrial life may look nothing like terrestrial life. So the question is, can we detect life as we don't know it? Specifically, extraterrestrial life may differ from the one on Earth in morphology and size. And even some life forms on Earth, like these coccolithophore that exist, I think, at least from the Cretaceous, is quite difficult to recognize. So eventual extraterrestrial life may present some shape that is too much exotic to be recognized from by our terrestrial eyes. Extraterrestrial life can also differ in biochemistry. Life on Earth is based on carbon and water, but on other planets it may be based, for instance, on silicon and methane. And this makes the recognition of eventual extraterrestrial life quite difficult, because its shape could be different from terrestrial life, and even chemical biosignatures may not match the ones we are used to on Earth. Here we have a very nice photogrammetry made by Girolamo Lorusso from the Natural History Museum of Piacenza that gives us a hint. We need for the search of extraterrestrial life a universal biosignature, that is, a biosignature that is ideally capable of detecting any type of life, regardless of its morphology and biochemistry. And meandering traces, like these scolicha, are approaching the idea of universal biosignature. Indeed, and this is a very well-known ichnological principle. Different organisms can produce the same trace. Meandering traces are produced by irregular echinoids. Worm-like organisms, like as shown by these Nereides from the Piacenza Apennines. We have even gastropods that produce meandering traces with the radula while they scratch algae. And probably the strangest example of meandering trace is provided by CD-eating fungi. So these slides show that different organisms produce the same trace, and this is a sort of tenet in ichnology. And if we bring this idea to the astrobiological side, we can acknowledge that meandering traces are not a signature of a specific organism or biochemistry. I can even expect a silica-based life form from an exoplanet to produce a meandering trace while looking systematically for food. Indeed, meandering traces are a record of an efficient foraging behavior. And if we find on another planet meandering traces, we don't even need to know what is the definition of life to acknowledge that this shape has been produced by a life form. So one of the big potentials of trace fossils is that they allow detecting life that differs from known life, and this property may seem trivial to us ichnologists, but it holds a great potential for recognizing exotic life forms beyond Earth. So, what extraterrestrial traces have we found so far? Now I can tell you that I lied to you in the first slides of the presentation. Actually, we found some traces on other planetary bodies, but these traces have been produced by humans. So it's not actually a revolutionary revelation I'm making you now. However, besides the blueprints produced in the 60s and the 70s by astronauts, we have other structures that look like ichnofossils. Some of these ichnofossil-like structures are these microfossils. They've been found in a meteorite collected in Antarctica, and these small structures have been produced on Mars more or less 600 million of years ago in igneous rock that pertained to the Martian surface. This Martian surface can come to Earth through a meteorite impact. This meteorite, Yamato 00593, preserves these micro tunnels that are enriched in carbon. That's another intriguing aspect, and their morphology looks a lot like terrestrial microborings. The microborings on the right have been reported by Ferretti and Ali in Silurian trilobites from the Alps, near the borderland between Italy and Austria. However, a recent study by McLaughlin suggested that these structures are not of biotic origin. I will not enter too much in details, but the explanation of McLaughlin et al. Is that these structures derive from an impact and subsequent hydrothermal fluid alteration. Another ichnologically intriguing structure has been reported by Nofke, and she described these sort of platforms that bear resemblance to microbially induced structures. However, the same author recently in 2021 retracted the potentially biogenic origin of these Martian structures. Then we have stick-like structure that have been not studied by Nofke, but by me and colleagues. Sorry, there is a mistake in the slide. And these millimetric structures from Mars have been produced about 3.5 billion of years ago on Mars. These structures are documented from the Gale crater, which is the landing site of the Curiosity rover from NASA. And within this crater, there are robust evidence of the past presence of water. We have mud cracks and even ripples. That's why a scientist hypothesized that the Gale crater was occupied by water when the stick-like structures have been produced. So this environment, clearly based on terrestrial standard, is quite compatible with life. And the structures themselves are morphologically very similar to terrestrial barrows. The fossil barrow in the center is a kind of Helminthoidicnites from the Carboniferous of the Pramollo Basin in northern Italy. And even these structures that I would say also they can be called Helminthoidicnites. They are barrows from the Cambrian of Sardinia that I'm studying with colleagues Pillola and Zoboli from the Cagliari University. However, I did not discover extraterrestrial life. If not, in this moment, I would be completely drunk in Sweden and being happy for being the next Nobel Prize winner. Indeed, these structures look like trace fossils, but they also look like sedimentary crack fillings. And the information we have at present don't allow us to demonstrate a possible biologic origin of these stick-like structures. However, the sole presence of these structures and their detectability using the rover tools demonstrates that we should keep our eyes open and check carefully every image that is sent on Earth by the Martian rovers. And in the third part of the presentation, I will show you what can be some tools to automatically detect ichnofossil-like structures from Martian imagery. And indeed, that's the third and the last section of my preservation, which is all about methods, tools, and also limitations of the astro-ichnological approach. One approach to study extraterrestrial life is focusing on extreme environments. And actually, more or less every ichnologist in his own or her career focused on extreme paleo environments. Because ichnology allow us to access to environments that otherwise would be very difficult to explore. For instance, this chondrites, a branched barrel, has been produced in low oxygen marine settings in the Silurian of what is now Sardinia. These exquisitely preserved Paludictium coming from the Piacenza Apennines, open a window in a deep sea setting. So it is by examining example like these traces producing extreme environments that we could imagine how extraterrestrial traces producing extreme environments would look like. So the astrobiological approach encourages us not only to look to the sky, but also to focus on what we have under our feet. Another tool that we can use to detect trace fossils on other planets is predictive modeling. Predictive modeling is a technique born in the field of archaeology that is used to predict the location of paleontological or archaeological sites in investigated areas. Me with colleagues from the University of Genova applied this approach to the Jezero crater, which is the landing site of the Perseverance rover. This approach is quite simple. The idea is to determine how suitable is a given point for ichnofossils. And in order to understand how suitable is a point, we looked at those characteristics that make the discovery of trace fossils easy on Earth. For instance, if I look to this picture that I took in Mongolia, these are Cretaceous fluvial deposit. I would say that I would not like to look for trace fossils in the smooth deflation surface. And even I would not like, I would not expect to find trace fossils on modern aeolian bed forms. Why? The answer is simple, because the rocky outcrops are completely covered by modern deposits. So there is no chance to find a trace fossil there. Trace fossils can be found in the uncovered bedrock. Indeed, here there are a lot of very nice like barrels. So I applied the same approach to Mars, and me and co-authors found that in the Neretva valleys there are quite suitable conditions for bioerosion traces. And I don't think that NESA looked at my study, but it is right here in the Neretva valleys that NESA scientists reported some structures that are at present one of the most convincing evidence for extraterrestrial life. In the predictive modeling approach I used, I did not only consider the percentage of superficial cover, but I looked at other variables that usually on Earth are associated to high bioturbation indexes. So I looked for low sedimentation rates, low hydrodynamics, and also based on the motto follow the water. I checked for those areas that on Mars were covered by water, because I need to point out that nowadays Mars probably lost its habitability after losing large water bodies on its surface. By summing using GIS system all these layers, I obtained a map where each pixel represents a score of suitability of bioturbation. In other words, in the red areas, I expect to find ichnofossils if bioturbating life ever existed on Mars. In the blue areas, the stability is a bit lower, whereas in the transparent areas, I don't expect bioturbation, because most of these areas are represented by igneous rocks. And from predictive modeling, we go to Commodore 64. I am joking. This slide is only to show how in 20 years we went from a very simple technology like the basic interpreter from Commodore 64 to artificial intelligence. Nowadays, every one of us can access to a very sophisticated tool like artificial intelligence that potentially could reveal or could help us to reveal potential extraterrestrial life. And I am trying to apply artificial intelligence to search for extraterrestrial and terrestrial life. Specifically, me and colleagues from CNR, Andrea Barucci, and from Evelyn Kustachev, we are for ichnofossils in one of the best Permian calderas in the world, the caldera of the Bolzano super volcano. In this caldera, we find not only ignimbrites, but also fluviolacustrine deposits that are very rich in ichnological evidence. For instance, this sample show fossil burrows, but also microbially induced sedimentary structures like this gas dome. According to our hypothesis, in the Permian, the sediment was covered by a microbial mat, and gas bubbles were trapped between the microbial mat and the mineral layer below. We are using examples like this one to produce an artificial intelligence model that is capable to detect ichnofossils when they are in the frame of the camera. Clearly, this technology has a nice potential on Earth, but presents a huge potential for systematically analyzing images from Mars and detecting eventual ichnofossil-like structures, like the stick-like structures I shown you before. How we are developing this artificial intelligence tool? Well, the reality is that the work is quite hard. Here, my collaborators Evelyn Kustacher, Bianca Miller, and Roberta Brands are taking pictures of bioturbated and bioturbated surfaces. We collected more or less 6,000 pictures, and we labeled them. So we told in the training phase of our artificial intelligence which photos contained unbioturbated images and which images contained bioturbated deposit. We did that for the whole data set, and hopefully this tool would allow us to spot interesting images from the millions of photos that have been sent and are being sent by the Martian rovers. So, in conclusion, I can present you the present, the past, and the future of astroichnology. The past is represented by the amazing potential of ichnology in the search for extraterrestrial life. Um, the potential is high, but I cannot hide the limitation of this approach. Clearly, astroichnology can be applied only in our solar system, where we can access to surface images of sufficient resolution of other planetary bodies. And there are also some limitations in exploring some locales, like the icy moons of Saturn and Jupiter, where the surface of the moon is covered by ice, so we cannot access to what's underneath. Another limitation is that we need benthic life for having potential extraterrestrial ichnofossils. Despite these limitations, I believe that the ichnological approach hold great promises for the discovery of extraterrestrial life, if any. The presence of extraterrestrial life is represented by a dialogue that at present is missing. Ichnology and astrobiology have much to tell each other, but at present there is not so much dialogue between the ichnology and astrobiology community. So I encourage to establish this dialogue that is much needed. And finally, the future of astroichnology is integrating it into future space missions. In 2028, we will have the Rosalind Franklin mission on Mars and the Dragonfly mission on Titan. And I would be really happy to look at the data of Rosalind Franklin and Dragonfly under an ichnological lens. So thank you very much for my attention. Here are my contacts. And obviously, if you have some questions, I'm here and I will be very happy to answer.
Thank you very much, Andrea. We have time for some questions, so you can open your microphones if you want, or leave your questions in the chat. Maxi?
I'm Mino from Piacenza, Italy. You heard me?
Yeah, I
Yes,
hear you. I hear
perfectly.
you.
Okay. Hello, Andrea. How are you? I think you're fine now. Okay. In Cagliari, you have began to study I think, in Paleozoic now,
Yeah,
and
yeah.
in Sardinia. And the beginning of life is the best that we can study for to found life on other planet, because we think that is simpler to find life in microbiological shape that not in macroorganism, as we are useful to found in on the earth. So I agree all you have explained us, and I hope that in a short time we can found and confirm your idea.
Mhm. Thank you for your comment, Mino. Yeah, yes, one of the let's say big conundrums in apparent conundrums in astroichnology is that on Mars we expect microbial life rather than macroscopic life. But the example of slime molds and even stromatolites, if we include them in ichnology, show us that microorganisms can form communities that can be detected macroscopically. I know that not every ichnology agree on including stromatolites in ichnology, but after all, stromatolites and microbial induced sedimentary structures derive from microbe substrate interactions. And besides that, there are also microorganisms like fungi that can aggregate and produce macroscopic trails.
Maybe we hope this that we can find this shape of microbial as stromatolites. Yeah, stromatolite.
We keep our finger crossed.
Okay, thank you.
You're welcome.
I have a quick question, Andrea. Thank you so much for your talk. Can you hear me okay?
Yeah, I can hear you.
Yeah. Do you think it's a catch-22 that we'll never recognize or interpret extraterrestrial traces on Earth until we find the same trace elsewhere, right? I mean, hypothetically, however small, there may be some problematic. Oh, let's say there's an extraterrestrial trace or made by something extraterrestrial on Earth, right? You could never publish a paper saying that I think this trace fossil, we can never come up with a trace maker. What if you interpret it extraterrestrial, right? Nobody will accept that paper probably until you find an identical trace somewhere else. I mean, it's more of a sort of philosophical, right, consideration.
Yeah, but I like a lot of philosophy, so the question
that's why I'm asking. That's why I'm asking you.
It's very welcome. I can say that meandering traces like the so-called helmintoids, narrators, if we found them or something like them on Mars, I think they are quite robust. They would be quite robust evidence for life. There are other trace fossils like helminthoid ignites or Gordia that can be mathematically interpreted as under the optimal foraging theory. So, in line of principle, we can say that similar morphology produced on other locales have been produced by derived from an optimal foraging strategy. Clearly, it would be great to find a trace fossils and also a biochemical signature. And may I just add a thing? That is usually in astrobiology, chemical signatures are believed to be more robust than morphological.
Andrea Baucon.
Io l'ho ascoltato durante il dibattimento.
Hello?
Yeah.
I think that there were some Italian friends there. So I was what I wanted to say is that usually biochemical biosignatures are thought to be more robust evidence for life rather than morphological ones. And I partly agree because we have a lot of examples of pseudo fossils that resemble body fossils. However, trace trace fossils are in a land in a Middle Earth, because they document behavior rather than the body of an organism. And the behavior, it's a property that is that pertains only to life. So I would say that as technologists, we should invest a lot of efforts on finding characteristics that allow us to demonstrate that a certain trace is 100% biological.
Yes. Thank you. Good luck.
You're welcome.
Good luck to all of us. Thanks.
Thank you.
Any other question?
I had a question. This was a really nice talk. I don't know how 40 minutes went so fast. Did you try adding meandering traces, mud cracks to your AI model? Because in the Permian photos you show the conglomerates and very straight burrows, but I was wondering if you added, the things that you consider, biosignatures. And also, like, are you planning to? Because I'm guessing also that it might be also quite interesting to try adding, more Ediacaran stuff to the whole AI model, which will hold, traces that could be more simple, and weird, and more simple mainly, I don't know if you are thinking on anything on that, or
Well, the Akaran idea, I think it's great. As concerns matrix, at present we did not include pseudofossils in our AI model. Just because we are trying we are starting simple to go more complex. But I can tell you that another model we developed but not still published on turbidite deep sea ichnological setting is capable to recognize ichnospecies of deep sea trace fossils with an 80% accuracy. And I would say that mud cracks are just another shape that can be detected from our model. So it's just to feed the artificial intelligence and hope for the results. And artificial intelligence holds also a great potential to detect pseudofossils, because we can try unsupervised classification. In other words, we can provide to the we can train our artificial intelligence with trace fossils, mud cracks, and other pseudofossils. And without giving any label, we can check if the artificial intelligence alone can discriminate between biological and non-biological forms. And once hopefully we find that artificial intelligence is capable to distinguish biological from non-biological shapes, and at that point we should try to explain the data. In other words, after training the model, we can check what are the characters that allowed the artificial intelligence to discriminate between life and non-life. And after that, we would have discovered the Graal of Ichnology. So the mathematical description of a biogenically produced structure.
Thank you, Andrea.
You're welcome.
Well, there are no questions in the chat. I don't know if anybody has any other question.
I have a lot of questions, but I just wanted to ask a short one. What is your opinion on the McLaughlin paper? That one that pre-described the micro kind of quote unquote burrows with fractures.
Well, to be very honest, I still did not form my opinion, because when I saw the first paper by White, and then there is another paper from another meteorite, NACLA, with the microtunnels, I thought, okay, these look very much like microborings, and there is even carbon inside. Then the McLaughlin paper placed some doubts in me. I need to read it better. But also, I would like a lot to focus on the so-called leopard spots discovered by NASA recently. They have been defined by NASA scientists themselves the most convincing evidence for potential extraterrestrial life so far. And here in Sardinia, we found on Cambrian surfaces very similar spots. So my plan for the future is using Sardinian data as an analog for understanding better Mars.
Well, thank you. Yeah, I wanted to have your honest opinion on that. But I really like the idea of, raising up Ichnology for astrobiology. Like it makes total sense when you put it that way. We don't have body fossils. The chemical biosignature doesn't always work, so trace fossil could help. Yeah.
Yeah. After all, even on Earth, the oldest evidence for life comprises stromatolites and microbially-induced sedimentary structures. So why not? And another thing is that we are the nightmare of body fossils paleontologists, because it happened to each of us. Many deposits have no body fossils, but they are full of trace fossils. So statistics is with us, I hope.
Thank you.
Okay.
You're welcome.
Well, any other question? If not, well, I would like to thank you, Andrea. It was a really great talk. And we are going to upload this talk on our YouTube channel next week. And as I mentioned before, this was our last talk for this year. So hopefully in February, we are going to start again with our cycle. So thank you very much for being here, and I wish you a great weekend.
Thank you.
Thank you.
Thank you. Bye-bye.
Thank you.
Bye-bye.